La fragmentación celular puede llevar a la desintegración completa de una célula y su eliminación durante procesos como la apoptosis. En este estudio, los autores descubrieron que el anclaje defectuoso del uso mitótico debido a defectos en la dineína y o en la miosina 1C acerca el ADN a la corteza celular. Esto desencadena la contractilidad de la actomiosina y genera fragmentos celulares. Utilizando un modelo embrionario murino, knockout, para el gen de la miosina 1C, los autores describen la formación de grandes fragmentos apicales libres de ADN, que esto ocurre durante la mitosis, y potencialmente compromete el desarrollo preimplantacional, lo mismo que ocurre con los embriones de fecundación in vitro.
Sí, el grupo de Maître describe que los cromosomas cercanos a la corteza celular provocan la acumulación de actina en el córtex, aumentando la tensión superficial de la célula, lo que desencadena un pinzamiento de una porción y la generación de un fragmento celular. Además, en este artículo los autores descubrieron que la vía de extrusión del corpúsculo polar está activa durante la mitosis en embriones en etapa de división celular. Esto se evidenció por la acumulación de la proteína CDC42, una proteína altamente implicada en esta vía, en la corteza de los embriones. Interesantemente, la activación de la vía de extrusión del corpúsculo polar en embriones normales no causa fragmentación celular, pero en embriones con mayor contractilidad y tensión superficial, la misma activación de esta proteína conduce a la generación de fragmentos. Así que, creemos interesante que gracias a este hallazgo, en futuros estudios se podría investigar la reducción de la contractibilidad celular o la activación de la vía de extrusión del corpúsculo polar en embriones humanos para mejorar el potencial implantatorio y reduciendo la fragmentación.
Thank you so much for being here. First of all, can you tell us briefly about your trajectory? What inspired you to work in developmental biology and specifically on early embryo development?
Yes, so I've been studying embryonic development now for since before my PhD even, right? So that's now quite some time, about 15 years or so, and so I got into it fairly early during my university studies, and so during my biology study, I had some dermatology biology classes, and then for my first internship, I joined the lab of Alfonso Martinez Arias in the Department of Genetics in Cambridge, and that was basically my introduction to dermatology biology. And Alfonso is very, very passionate about his research, and he shares it in such a unique way that it's very communicative. I wouldn't say that I would not have fell in love with dermatology biology without Alfonso, but Alfonso was definitely, definitely played a part in that.
Good. So we would like to know you a bit more. So for that, we would like to ask you if you could go back in time, what science advice or what career advice would you give to your younger self?
Yes, so I thought a little bit about this. So my answer is fairly technical, actually. I feel like I would definitely advise my younger self to learn more about coding. I could code a little bit, but I didn't spend enough time on this, and I realized how important it is in today's research, and I feel like I would be much more efficient, and I could do many more things if I could code a bit better. So this is definitely one thing I would advise people to learn and to pick up. It's it's everywhere, and if you can code, you can do so much more.
That's true.
I think we can all agree to that, definitely.
Yeah. Sometimes when you are doing something very repetitive, you think like this could be code and I could be much faster.
Definitely.
And it takes time, right? You need to practice and you need to use what you learned, but it's well spent time.
Sure.
I think that bioinformatics will be a must quite soon. If it's not already a must in our field, it will be for sure a must in a few years' time.
Yes.
So, Jean-Luc, going to your work, which is impressive, actually, this is really impressive. In the text, you literally say that serendipitously found that the loss of myosin 1C causes a partial fragmentation in mouse embryos. And it's funny how, despite the rigor that we are asked for in science, some of the best scientific findings are discovered by serendipity. So, how did you manage to find this in the lab? And also, do you remember the day that you actually discovered this? What did you think? What did your coworkers say? How did you react?
Yeah, so I mean, we all know, right, that many discoveries are very much due to luck, right? And then you need to be prepared to catch them, right? And this is not the first time that something which leads to a paper was discovered by luck in the lab. And sometimes you try to write a paper in a way that this was well thought through, and you discovered this because you had a great insight and so on and so forth, right? And so this is not the first time. Now we try to rationalize the fact that of course we would have to look into this gene in particular to discover about fragmentation, but there was just zero link, right, between this gene myosin 1C and fragmentation that we could draw. So we just decided to just be transparent and honest and just say, well, this was serendipitous, right? But that's because we couldn't find a rational way to connect this gene to any fragmentation behavior before. And then so the way we bumped into this is that so Diana Pelzer, the first author of the preprint, so she started knocking out several genes using CRISPR-Cas9, and so those genes so these were like cytoskeletal and adhesion-related genes, which we suspected could be involved in pre-implantation development because those genes were expressed, but there was no known phenotype for those genes. And so she developed a pipeline where she would knock out a gene in the zygote, right, and then image the development of those embryos. And so she could screen through something like 20-something genes, right, in about a year or so. And then one of those genes was myosin 1C, and we did not immediately got interested in fragmentation. What we were interested in the beginning is a more rare phenotype that those mutants have, which is the fragmentation during the interphase. So in the preprint, we see that fragmentation is mostly happening during cytokinesis, but occasionally what we can see is that there's a fragmentation there's a fragment that forms during the interphase, and this is a result of some kind of contracting ring which forms and then just pinches off a piece of the cell. And we suspected that this ring was related to the formation of the apical domain, which appears at the 8-cell stage during implantation development. And so we were very interested in this ring and how this ring could bulge and then pinch off. And this was just a very rare event, which was very difficult to study. And then we noticed the fragments and we thought, okay, well, actually, this is not just how those fragments form. It's not just with this ring that constricts during the interphase. It's mostly during cytokinesis. And then when we looked in the literature, we saw that actually the way that human embryos fragment was not very well described. There were some proposals, but we felt like those explanations were maybe not correct or a bit too far-fetched. And so that's when we decided to shift gear and say, okay, we stop looking at this rare event and we look at this other rare event, which is fragmentation.
Wow, it was impressive explanation too. And as my peer Rafa, I would like also to congratulate you because of the way that the preprint was written. It was like a story. It was very easy to read, and for me it seems a very innovative way to write too, on a scientific paper. And now,
on this,
well, yes.
I would just like to say that I think, to some extent, this is what we want, right? I mean, we, as scientists, we are interested in the truth, right, and in discoveries, but we are not interested in a list of facts. We want to be told a story, just like children.
Sure. It was very easy to follow.
Thank you.
So now we would like also to go more into the content of the embryo fragmentation and take it to our field of assisted reproduction. And we would like to ask you, as you mentioned in your work, embryo fragmentation has not always been observed in other mammalian embryos, for example, in mice. Do you think that this is a factor contributing to the low fecundity of human species? Because we in humans see a lot of fragmented embryos.
Mm-hmm. Yes. So, I mean, I'm sure you know better than me all the different problems that human primitive embryos can have, and fragmentation is one of them. So I would not, I would not be able to tell you, how much fragmentation is contributing to the low fertility of our species, but it's definitely not helping. That's for sure. Whether, I mean, based on what we find in this paper, we think that this is related to some extent to defects in the positioning of the mitotic spindle. And if the mitotic spindle would be defective and missegregate chromosome without fragmentation and create a mosaic embryos, I'm sure this would also be bad even without fragmentation. Now the fragmentation doesn't help for sure. And I think it would be, I think it's a very interesting question, because like from all these problems that we see, and everyone is claiming that this problem is the main problem, I mean, how much do they contribute? How much does each of them contribute to our weak fertility? That would be very interesting to look into.
Indeed. Indeed. And actually, just with regarding the answer that you just provided, do you think that ovarian aging may be associated with a hyperactivated polar body extraction pathway, and thus leading to an increase in human embryo fragmentation in older women?
So this we didn't look into at all, right? So the effect of aging, and my knowledge of the aging field is not so strong. But what I know is that during aging, our tissues globally become stiffer. This is a general feature of aging, and in a different study that we have also submitted to bioRxiv early last year, where we measured the mechanical properties of human embryos, what we found is that human embryos are much more tense than mouse embryos in general. We did not explore the aging factor. But we noticed that those embryos were just very, very tense compared to the mouse embryos. And if with aging, the embryos, and presumably the oocytes, which gave rise to those embryos, are becoming more tense with age, this could impact the fragmentation process. Because what we found is that the fragmentation comes from an ectopic contraction. So the cells contract the actomized cytoskeleton in a place where they should not. In the mouse embryo, by overactivating this actomized cytoskeleton, we made the cells more tense, and this led to fragmentation per se. So the aging factor could also kick in at this point, because if age makes the oocytes more tense in embryos which are already more tense, maybe that's just making things worse.
Okay. Thank you.
Thank you. Well, so you have already mentioned it, but let's see if now we can have it more clear. Defects in the spindle anchoring are related to aberrant chromatid or chromosome segregation. and therefore may explain embryo aneuploidy or mosaicism. So, taking into consideration that findings, do you think that embryo fragmentation could be associated with mosaicism or aneuploidy?
Yes, I mean, definitely, because what we see is that not all fragments contain DNA, but about 20% of them do in the mouse mutants that we characterized. I do not remember the figure for human embryos, but the figure is there somewhere. Some fragments do contain chromosomes. This has also been studied in more detail in a study in monkeys, in macaques. And they also found chromosomes, which would be trapped into the fragments. And so we think that the reason the chromosomes get trapped into the fragment is because in the first place, the chromosomes were the one instructing the surface of the cell to constrict and to form this fragment. And so this is definitely going to contribute to cells losing chromosome. And so the way cells can lose chromosome is, first of all, they could lose them from their nuclei. So you would have the main nucleus with most of the chromosome and then a micronucleus forming somewhere. That's already a problem. But then if this micronucleus is forming even in a closed entity at the surface of the cell, that makes the cell aneuploid. So that's definitely contributing.
That's Rafa. That's very interesting for us in the clinical practice, right? So it was like you were describing the major part of our embryos normally. So thank you so much.
Thank you for that. And again, cell fragmentation is especially evident in cleavage stage embryos. Could we infer that after embryo compaction with the formation of the morula on day 4 stage development, the polar body extrusion pathway may become inactive?
Yes. So when this aberrant usage of this polar body extrusion pathway stops is something which we don't know for now. As far as I could see from, so by looking at other papers in the literature, what we can see is that actually at the 16 cell stage, there are still some pictures of cells dividing in the mouse embryo where you see that the chromosomes come close to the cell surface and induce the formation of an actin cap. This has been reported, and it's not, it's a completely different study, so it's not from our group. And so we suspect that this could still be active at this stage. Whether it's active in all cells, I do not know. What I think could be an important factor in reducing the formation of those fragments as the cleavage stage progress is that, as I mentioned to you, the fragmentation results from this aberrant signal, but also from this hyperactivation of actomyosin contractility and this high tension. And so what we could measure in the mouse, and that is again a different study from the lab, which came out last year, is that during cleavage stages, the tension of the cell is decreasing. It starts very high in the mouse at I think about 1,500 piconewton per micrometer, and it goes all the way down to 200 at the 4 cell stage. And so cells are getting less and less tense, meaning that when the signal from the chromosome to trigger this fragment comes in a zygote, it has a very responsive and very strong actomyosin cytoskeleton. When it does the same at the 4 cell stage, it has a much weaker response from the actomyosin cytoskeleton. So maybe the signal is still active, and we see evidences of this being still active at the 16 cell stage, but the response from the cell is weaker because it contracts less. So maybe that's it, but I don't know.
It does make sense.
It's an explanation.
Totally, yeah, that makes sense. So, well, we have arrived to the last question from our part. That will be trying also to go to the IVF field where we work on. Do you imagine any mechanism or pathway by which in the IVF lab we could avoid aberrant embryo fragmentation? Or maybe during the clinical intervention with the ovarian stimulation or something like that?
Yes, so we thought about this. We are a lab of biologists, but we work together with clinicians as well, and we definitely thought about how we could translate this knowledge into the clinic. And so there's one target in particular which I think is promising, and we are currently not trying anything with it, but I think that'd be worth it that someone tries. And this target is CDC42. So CDC42 is a small GTPase, which is very important for the embryo and for the oocyte to organize the actin cytoskeleton and organize the apicobasal polarity of the embryo. So CDC42 is very active during meiosis, and if you interfere with CDC42 during meiosis, then polar body extrusion will not occur nicely, or it will divide. The egg will divide symmetrically, which is bad. So you don't want to interfere with CDC42 before meiosis. But after meiosis, CDC42 is not required until the 8-cell stage. So at the 8-cell stage, that's when apicobasal polarity is formally established in the mouse embryo and most likely in the human embryo as well. And so between the polar body extrusion during meiosis 2 and the 8-cell stage, it is very possible that CDC42 is not so important, except for the extrusion of those fragments. If we block CDC42, we see that the fragments are not forming as nicely. This actin induction by CDC42 is not happening anymore. And so that could be a very good target. And there are several chemical inhibitors of CDC42 which exist, and we have not tried them, but they could be a target.
Wow.
Very interesting indeed.
Yeah.
Actually, we have come to the end of our questions and to the end of the interview. I think you have opened a magnificent way to explore embryo contraction and tension. Thank you very much, Jan, for your time.
Thank you for your very good questions. Thank you very much. And thank you.
We have learned a lot. Yeah, we have learned a lot. And finally, of course, we encourage you to following with this research in particular that for us is that interesting.
Thank you very much. Thank you very much for your time.
Thank you.